Hey you. Yes, you. Have you been sitting in your room at 4:30 in the morning and suddenly ask yourself, what the heck are those little worm things at the bottom of the nutrient cycle? Well, you young, dastardly handsome biology student, those are nematodes. Now, you may be asking yourself, a nematode? What's that? A nematode is a special microorganism found in the environment that plays a crucial part in our nutrient cycle. They help crop growth and start the food chain, providing nutrients to the surrounding dirt and water. Nematodes have many variations. This one feeds on bacteria. This one feeds on fungus. This one's a cannibal, and this one feeds on your brain. But don't you worry, you won't have to worry about that one. But wait, you may be asking yourself, you said they grow in soil. Does this mean that they grow in artificial soils too, like fertilizer and potting soil? And if so, do they grow more efficiently in an artificially nutrient rich environment? Well, I hypothesize, and so does a similar study done at the University of Missouri, that if the soil is already artificially nutrient rich, then the nematodes will be more abundant in said soil. Oh, yeah? You may be thinking to yourself, well, then prove it to me, Mr. Narrator. Do an experiment with a simple and easy to digest format that can be recreated for later in preferably 2 to 4 minutes with language that a non STEM major will be able to understand. That's a rather oddly specific request, but we'll do it. First things first, we have to collect our samples, 4 to be exact, from 2 different locations, one with potting soil and one without potting soil. These will be our control and experiment groups. Then, prepare your samples. Fold them into tiny little burritos and then smack them into 50 milliliter Falcon tubes and fill up with 10 milliliters of water. Then, let sit for 24 hours. Now that our nematodes have sat for 24 hours in water, it's time to extract them. Take a pipette and extract up to 5 milliliters of the water and put it into a watch glass. Now, let's put our little nematodes under the microscope and you can watch the miracle of life. Oh, nope. Yep, they're all dead. You killed them. You monster. It's your fault. You did this. They're dead because of you. They had families, tiny nematode families, and you killed them. They're all dead. Have you no shame? Their blood is on your hands, you murder. Now, if this experiment was done right, you should be seeing lots of dancing little nematodes in our watch dish. Now it's time to count the living nematodes in each of our samples. Be sure to record down the number you see and mark down if you see any variations or different types of nematodes in your sample. This should give us an idea of how well the nematodes survive in each environment. Now, in our experiment, we had some rather unexpected data that showed that nematodes in fact were more abundant in a natural soil than in an artificial one. According to our data, a natural soil will have more nematodes in it than a non-natural soil. Now you know, you young, brave, budding scientists. Now go out. Go experiment for yourself.